These notes are on protein synthesis, and they're going to include information on RNA, transcription, and translation. So if you remember back to the notes that we took in 5.1, we mentioned RNA briefly, and we talked about that it has 3 different types. There's mRNA, which is messenger RNA, there's tRNA, which is transfer RNA, and there's rRNA, which is ribosomal RNA. So these are the 3 types, and really we're going to be getting into these 2 the most. Let's write a little something here about ribosomal RNA, since we're not going to really be looking at that too much later on. This rRNA is structural in ribosomes. So we find it making up the ribosomes. mRNA is made in the nucleus from DNA, and the section of DNA that codes for a protein is called a gene. So a gene is just a section of DNA. The base coding rules for DNA to mRNA are A to U, T to A, C to G, and G to C. So just like base pairing rules in DNA, we have the same sort of thing matching up, only notice that we have U replacing T. There are, and please write this, no T's in RNA. And that's how you can basically, one of the ways you can tell the difference. Okay, so this is an example of how this works. We have a sequence of DNA, and we have an A, T, C, which codes for UAG, and when you're looking at mRNA, they're read in groups of 3 bases, and that's called a codon. So you see CCC gives us GGG, and that's a codon. So this would be 1 codon, this would be 2 codons, this would be 3 codons. Each codon codes for a specific amino acid, and then the amino acids link together to make a protein. So a group of 3 letters gives us an amino acid. And then another group of 3 letters gives us another amino acid. And then these are going to be bonded together by what we call a peptide bond. So make sure that you write that. Amino acids bond together by peptide bonds, which are just strong covalent bonds. Okay, the next thing that we want to look at is this chart. This is how you figure out what the mRNA is coding for, which amino acid. And it's called the amino acid chart. So for example, if I have an mRNA codon that reads AUG, I go to my chart and I find the section labeled A, which is right there. I then know that I'm somewhere within this quadrant. The next letter is U. So now I'm in the A and I go down to the U area. Fix my line a little bit. So now I'm in the U area. So I went A, U, and then my next one is G. So I come down here to G and I find that it stands for methionine. And we would abbreviate that MET. And that's my amino acid. So the process of taking DNA and copying it into mRNA is called transcription. So that's DNA to mRNA. And this happens in the nucleus, and it occurs at the gene necessary for the protein that needs to be made. So not the entire DNA, and that's important to know. We're not taking the full DNA and transcribing it into mRNA all at once. We're just doing gene by gene as we need them. And this is a picture of that. And we're going to be going over this whole process step by step in the next video. So this is just sort of an overview for you of how this is happening. This big white thing here is our enzyme. Remember, it's always going to involve an enzyme to facilitate what's happening. The other process that is involved in protein synthesis is the process of making an amino acid chain from the code on mRNA, and that's called translation. So we're translating the mRNA into the sequence of amino acids, and this occurs at the ribosome, and each codon is translated into an amino acid. And this is a picture right here. You see the mRNA leaves the nucleus and it comes out here, and this right here is our ribosome, and this is our mRNA long strand, and then this is another type of RNA called tRNA that brings our amino acids, and we're going to be going over this in the next video as well. So transcription and translation together make proteins. This is protein synthesis, and it starts in the nucleus and it ends at the ribosomes. So the next video will go through the steps of transcription and translation together.